Here we go. We're on part 7 now, considering muscle histology, and we're now going to move on to the physiology and consider what is the homeostatic metabolism needs of muscle tissue. And we're going to come to this page right here and look at muscle metabolism, and we're going to consider the needs of the muscle. Of course, we have the action potential, which requires that we have sodium and potassium, and of course we have the initiation of contraction, which requires calcium. So we have certain electrolytes there. Okay, and then the next detail here is that we need to have ways of acquiring ATP. So for the energy of the muscle, we need to acquire ATP. Let's take a look here. We have direct phosphorylation of ADP by creatine phosphate, and this is based on the idea that we're going to, during rest or sleep, part of the biosynthetic pathway is to phosphorylate creatine in muscle cells. Okay, this is a significant factor in muscles that are used for strength or quick action, and it doesn't require a great deal of metabolism. Once you have the creatine phosphate, you can quickly phosphorylate ADP to get ATP, which is the molecule of energy currency. Okay, so the number 2 pathway then is anaerobic glycolytic pathway. So we're going to take glucose and break it down into a couple of lactic acids, and from that simple breakdown we're going to get a couple of ATPs out of the deal. Now we can go further with that if we're slowing down enough to be aerobic. If you're anaerobic, it means you're going all out. You're not able to actually speak clearly because you're using your wind to support your muscle activity. But if you slow down enough to actually run and be able to speak at the same time, you're probably being aerobic, at which point you're entering the mitochondrion. Okay, and the mitochondrion has Krebs cycle, has oxidative phosphorylation, and we're going to make actually 38 ATPs out of every molecule of glucose. That's a lot of ATPs compared to just 2 for being anaerobic. So let's take a look at the diagrams over here. We have a comparison of 3 different pathways that I just spoke of. We have, in this particular pathway, we have the direct phosphorylation, it's referred to, and we're going to already have prepared our creatine in this regard during sleep or rest. We're going to phosphorylate it, so it's all set to go. And then during activity, we're going to take the ADP that's available in the cytoplasm, and we're going to quickly just flash and add a we're going to phosphorylate the ADP to ATP and have it available. Eventually, when we're resting, we're going to have to use some ATP to make new creatine phosphate, so that's the recovery. Then we have glucose. For quick energy in the cell, we're only going to go this far. There's no oxygen available because we're sprinting or we're doing something quickly, so quickly or with such great strength that we're only getting to the lactic acid. We don't have enough oxygen to take the next step. And we're going to get 2 ATPs out of 1 molecule of glucose. If we slow down a little bit, we can have a little bit more endurance, and not only can we go to the pyruvic acid, but now we can actually enter the mitochondrion. The mitochondrion has coupled pathways. It's coupling Krebs cycle with oxidative phosphorylation. We'll have to talk about that in some detail. That's a cell biology, biochemistry issue. Okay, but the deal here is that we're going to make actually 38. Ignore the biology situation here. The biochemistry is we're not dealing with membranes. Okay, this is an approximate number. It could go anywhere from 28 to 32, depending on how you wave your hands and what membranes you consider. But if you're just in a test tube and you break down glucose, you get to 38 ATPs. And that's the number you actually have to know for your board exam. So board exam, entrance exam, 38, 1 molecule of glucose. Keep that in mind. Okay, you heard it here. Okay, so that's quite a bit more, and it means that you have oxygen available. So here we're actually having oxygen available. It pulls everything through. There's some place for everything to go. Without oxygen, it's just stuck. We're stuck at pyruvate. We're going to shift over to lactic acid, as noted in this situation, and basically we're going to experience what's called the burn. Okay, that means you sprint until it hurts, or you lift weights until it hurts. Here we're actually going to back off just a little bit from the anaerobic so that there's oxygen, and we keep right on going. And what are we mostly burning in this? We're actually mostly burning fat. Probably 65 to 70% of the ATP is not coming from glucose. Actually, it's coming from fatty acids. So we're only using the glucose to support the burning of fatty acids. So they sort of only reference that sideways over here. Okay, but when we get to actual chapter on metabolism, which is over here later in the book in Biology 20b, then we actually learn a few things about how does this really work. Okay, so here we have acetyl-CoA, and actually this can come from fatty acids. And it can be just supported in the Krebs cycle. Glucose actually creates the precursors in the Krebs cycle for metabolizing fatty acids. Okay, and then we send linkage molecules, B vitamin molecules, riboflavin and niacin over here, and we actually create most of the ATP. As you can see, there's substrate ATP here, substrate ATP here, 2, but the real major contribution comes from oxidative phosphorylation to get our 38 ATPs. Okay, so that's going to be coming to you. Stay tuned, and we'll get to Biology 20b, and you'll have even more information. Okay, so that's that deal right there. Okay, now we have to consider the idea of recovery here, but first of all, we're going to take a quick look at activity. Here we have somebody who's actually going to do a short duration exercise like running the 100-meter dash. Okay, and so in the first few seconds there's available ATP. Then we're going to use up the creatine phosphate. Okay, and then if we keep going now, of course we can do a quarter mile, we can do a 400-meter run, and some people can actually sprint for that long, in which case we start to use glycogen directly as glucose, and we get that substrate ATP from the deal. But we're only going to be using glucose, nothing else, creatine phosphate and glucose. This is an anaerobic situation. Over here, we consider prolonged duration exercise. We're going to go for hours. All right, so we're running a marathon here. This woman might run the marathon in 3 hours or 2.5 hours. So in order to do that, you're going to have to burn fat. There's no other way to do it. You don't have enough glycogen. She also can't sprint. She can't waste her glycogen. If you're going to run 26 miles, the only way you can possibly survive for 26 miles is to run a very, very, very steady pace, using your breath well and not ever sprinting. Okay? Sometimes psychologically people try to psych each other out during these races and try to do a quick little run up ahead and stuff like that, break away. But breaking away is, how shall I say, not a brilliant idea if you're actually going to have your best time. We have recovery. Recovery is important. At the end of all this, when we're resting, we have to have adequate oxygen back into the myoglobin, so we have to replenish the oxygen. We have to remove the lactic acid, metabolize it back into pyruvic acid, and go into the mitochondrion. We have to replenish glycogen because we only have storage for 1 day, and then ATP and creatine phosphate need to reverse themselves, and we're going to reestablish creatine phosphate.